So they say that we have microplastics in our lungs. You may not be surprised at this point to learn that the Guardian was the first to report this to the general public, again, based on a paper that hadn't even yet been published but had been accepted for publication. They state that particles were discovered in 11 of the 13 tissues that were examined. And there are a few other statements in here that I'm not going to pick out for you right now, but I will come back to some direct quotes from this paper and talk about the reality of the situation. So this is a scientific article. It's in Science of the Total Environment 2022. They take some lung tissues from patients undergoing surgeries and digest them and filter and recover some particles, which are then characterized as either microplastic or other. And they do state that they have 11 of 13 lung tissue samples that have some microplastics. However, only one of them is above what's called the limit of quantification. You may recall from the blood paper that that was the threshold that was used to say whether or not particles were found in the blood, was limit of quantification, which is a higher bar than limit of detection. I'm not sure why they went to a limit of detection for this description, probably because it sounds more alarming than saying one of the 13 was shown to have microplastic particles. But really, by the limit of quantification, there's only one sample that was above that. Everything else is just, there was enough there to see it, but not enough that they could say for sure how much it was because it was too close to the baseline, too close to the control experiments. There's a lot more in this article, but as I like to do, I would prefer to go to the data. And so this is a summary of their 13 data points in an Excel sheet. These data points are corrected for background. That's why the microplastics found on the y-axis in some cases are 0, and in one case even less than 0, because there are more microplastics in the controls than in the sample itself. But in general, there's somewhere between 0 or -2 and 7 particles found after correcting for the control experiments. And I started with this analysis by just plotting the microplastics found versus the size of the tissue sample. This is the analysis that I used in the breast milk study to demonstrate that it was unreasonable to expect that the microplastics actually came from the breast milk itself, because in that case it had a slope very close to 0 and was in fact negative. And there was a clear alternative source of the microplastics other than the breast milk, and that is that it came from the surface of the skin itself. So in this case, the slope is a little bit positive, but it's still well short of what's expected based on a random distribution of particles across the tissues, the tissue samples. And if you want to know more about this kind of analysis, I went into a little more detail in the breast milk case. But there are 3 big differences between this data set and the data set in the breast milk case. 2 I've already mentioned. One is the slope in this case is in fact positive. The second is that, at least for me, there's no obvious alternative source of the microplastics other than from the lung tissues themselves. I couldn't see anything that is not taken account of by the way they do their control experiments. So that doesn't mean there isn't something. There's always a chance that there's something that they didn't think of and I didn't think of, but there's no obvious alternative source for the microplastics. But I think the most important thing is that in this case, there is an a priori reason to expect differences among these samples. And that is that some of them were taken from the upper region of the lungs, as shown by these orange points, some from the middle, as shown by the blue, and some from the lower regions, as shown by the black. So if you separate them out based on where within the lungs the tissue samples originate, then you can see a pretty solid case for the middle and lower regions. So let's look at these data in one more way. I want to walk you through this Excel spreadsheet, which again contains all of their data, so you can understand a little bit about how they account for the controls and how those controls influence the conclusions of this study. So they divided into 2 batches, and for each batch, they do a number of procedural blanks, and that just means that they do exactly the same procedure, but they leave out any tissue sample. And what they found is for batch 1, in one case they had 4 microplastics, in the other case they had 0. And then batch 2, they had 0 and 0. In addition to that, they do a lab blank, and that's essentially just an open container that can accumulate any particles in the laboratory where they're doing the work that may happen to settle into the experimental sample, or in this case in the blank sample. And then they correct for both of those by subtracting the average of the procedural blanks and then the lab blank specific to that particular sample. So for example, here you have 8 microplastics, you subtract 0 for the lab blank, and then you subtract the average of these 2, which is 2. That gives you an adjusted number of 6, and that's what's used in the analyses. And that's the same for all of these 13 samples throughout. Then you can see these are the results summarized with 90% confidence intervals. I think this gives a good snapshot for what is observed in this case. So in the upper areas, there's really no good case for any microplastics being found there. In the middle lung areas, this 90% confidence interval for the most part is above 0, so it does seem likely that there are a few microplastic particles in the middle lung areas. But then the lower lung areas is where there's much stronger evidence, above 1.5 microplastics per average per gram, with the 90% confidence interval well above 0. I do want to point out, though, that these confidence intervals and these results are all assuming that the control experiments were done correctly and subtracted correctly. So that assumes that these adjusted numbers are correct. Okay, so all of this is a reasonable way to do the experiment, but it's a little concerning that they only do 2 controls per batch. Okay, and let me show you why. So again, you have 4 here and 3 zeros. Well, what if this had been a 0? It wasn't, but what if it had been a 0, which is not unreasonable to think that it could have been? Well, in that case, you would have said in the upper region, now all of a sudden most of that 90% confidence interval is above 0. So you would have said, yeah, there's probably a little bit in the upper region, but the lower region now, it goes all the way up to 3. So your estimate for the concentration of particles in the lower regions of the lungs is doubled. And that's all just based on one control experiment, which could have been measured differently. So now let's look at the opposite case. Okay, let's say what if instead of both zeros, this was a 4, which is what they measured. Now let's say this is a 4, which again, it could happen, right? I mean, it's not unrealistic to think that one could have been a 4 since the other one was a 4. We'll see how that completely changes the results of your study, right? In this case, you would have said, yeah, it really looks like there are no microplastics in our lungs at all. Maybe a little bit in the middle section, which would have been a very confusing result. But even the lower region now, you would say, no, there's no microplastics there. So this one control experiment makes a big difference if you get a little bit different number there. And this could have been easily alleviated by simply doing more blanks, right? So why not do 5 or 10 blanks there and have a little more confidence in your results and not have them so dependent on this one data point. Having said that, this is what they measured. They measured 4 here and 0 there, and they reported all of this, and they analyzed all of this. And so, you know, based on the data they have accumulated, their analysis makes sense. And the most likely interpretation of these data is exactly the way they interpret it. So it really seems like, yeah, there are microplastics in our lungs, not in the upper regions, probably some in the middle regions, and probably more in the lower regions of our lungs. Okay, let's go back now to the original Guardian report and analyze a little bit about what was said there. Okay, these are some direct quotes from the Guardian article, particles discovered in tissue of 11 out of 13 patients. Well, as we discussed, yeah, that is what was stated in the article, but that doesn't account for some of the particles that likely originated from external sources based on the fact that a number of the controls also had particles in them. So this is almost certainly not correct that 11 out of the 13 samples had microplastics in them originally. I think it's safe to say or fair to say that about 9 of the 13 lung tissue samples had particles in them because there were 9 positive values after subtracting the controls. And then I would also mention quantifiable in 1 of 13. And this may be a little bit nitpicky, but why not go ahead and be accurate, right? It doesn't cost you anything. Next is a direct quote from one of the authors. We did not expect to find the highest number of particles in the lower regions or particles of the sizes we found, which I think means large. I'm fine with that. They didn't expect it. I didn't expect it, honestly. But thinking about it a little bit more, I would guess that our upper and middle regions of our lungs are probably exposed to microplastics in a much higher number, but they just don't stick there because the airways are so large there. So we just inhale and exhale, and it's only those particles that get down into the lower regions where the airways are a little bit more constricted and they can get stuck in there. So I'm guessing that's what happens. I'm very curious whether those particles in the lower regions stay there, and if so, how long. Are they just there until we have a really good cough and then they come back out, or do they really get stuck in there and we just can't get rid of them? And that I don't think anyone knows. Let's go back to some statements from the Guardian. So, workers exposed to high levels of microplastics are also known to have developed disease. That sounds concerning. They don't give any reference for this statement, and they don't say what the disease is. My best guess is that in this case they're talking about flock workers' lung. People working in the textile industry were exposed to high levels of microfibers in the air, and they breathe in enough, they really get stuck in their lungs, and that can cause interstitial lung disease. This has been essentially taken care of by engineering controls that, really this is kind of true for any airborne particles. Think about asbestos, for example. Think about silicosis, for example. Long exposures to dust, right? Any airborne particle in high concentration with chronic exposure, you're going to have this kind of a problem. So yeah, if people are exposed to high levels of microplastics over long periods of time without the appropriate precautions, you can get some lung disease. Let's take a look at this last statement, because I've seen something similar to this in several articles, including scientific articles. Microplastics cause damage to human cells in the laboratory, and air pollution particles are already known to enter the body and cause millions of early deaths a year. Now, if you read that quickly, you're going to think that microplastics are causing a lot of human deaths. That is not true. And this is just a guilt by association. Now, what does cause a lot of deaths is air pollution. But it's the boring air pollution that we've known about since the 60s and 70s. It's things like tailpipe exhaust, volatile organic component, and heavy metals. These are the things that have been shown scientifically to be the air pollutants of concern. Microplastics are present in the air generally in the 1 to low 10s of particles per cubic meter. Tailpipe exhaust, if you do some conversions and make some assumptions based on some reports from cities, for example, are in the millions, maybe higher, depending on what assumptions you make about particle size and shape. But there's certainly several orders of magnitude higher in abundance in ambient air, right? And that's what the real problem is. There have been some other studies that have shown VOCs and heavy metals to be especially bad actors. There's really no evidence that microplastics are particularly bad actors. They are present. We do inhale and exhale them, but the people who study the real air pollution problems that are really leading to deaths, the microplastics they're just not prevalent enough to even figure into the analysis. So this is a really misleading statement, and it appears in various forms in a lot of papers. Okay, so bottom line is it does appear that we have microplastics in our lungs. I wish the authors would have spent a little more time on method validation and on method development. And I wish they would have done some more control experiments to give all of us a little more confidence in their numbers. But the data that they have and that they have reported are indicative of microplastics in our lungs, and that seems to be the case. Thanks for listening. There's more to come.